Hello and welcome to the Restore Wellness Podcast. We have a very special crew with us today. In the studio, we have Jimbo Collins.
How's it going?
And Keith Tenenfeld.
Hello.
And on the Zoom call here, we have Dr. Thomas Seyfried. Welcome, doctor. Thank you so much for coming on.
Well, thank you very much. It's a pleasure to be here.
So you've done some things with cancer that are definitely, like you were saying in our earlier discussion, different than the standard care plan that they have. Give us a little bit about your background and how you got onto this path that's now in the forefront of what's going on with cancer research and treatment.
Well, when we started this, you have to realize that my training is in lipid biochemistry and genetics. I have my Ph.D. in genetics from the University of Illinois. And then when I was at Yale University for 9 years in New Haven, Connecticut, I was working with the late Dr. Robert Yu, leader in the field of ganglioside biochemistry. These are complex glycosphingolipids. And we were always asking questions about what the function of some of these molecules were. And we were using different developmental programs as well as cancer. But it wasn't, none of this was done to manage the disease at all. And while I was at Yale, their department of neurology was focused very strongly on the field of epilepsy. And so I was mapping genes for epileptic seizures in mice with the hope we could define something in humans. At that, I try to write a grant for on ketogenic diets for epilepsy, which is where ketogenic diets have their biggest impact clinically, is in the field of epilepsy. Little kids all over the world are using ketogenic diets to manage seizures. This is one of the top ways of treating children with epilepsy. So I wrote up a grant for ketogenic diet for epilepsy, and they said to me, nobody's interested in that anymore. So I didn't get the grant. But then when I was at Boston College, continuing my research on ganglioside biochemistry and angiogenesis and cancer and this kind of thing, one of my students, Jim Abrams, who just passed away recently, made the Naked Gun movies from Hollywood, Airplane and these kind of funny movies. Yeah. So I knew Jim really well. His son Charlie had epilepsy and it wasn't responding. So he made the movie First Do No Harm with Meryl Streep, and they started the Charlie Foundation, which was designed to use ketogenic metabolic therapy for managing seizures. So I had already a big epilepsy program at Boston College with preclinical studies. We were trying to map genes. And then finally, the problem at Yale was that they thought drugs and all this other stuff would be the better way. But then when Jim started to build the intention around ketogenic diet again, that was kind of left. It was first developed in 1921 at the Mayo Clinic by a guy named Wilder, who said you can manage seizures if you do water-only fasting, but obviously you could only do that for a short period of time before you enter into starvation. So he developed this, unpalatable ketogenic diet that seemed to really work in blocking epileptic seizures. The mechanism by which ketogenic metabolic therapy blocks epilepsy is still debatable. All we know is that you have to keep blood sugar down and keep it steady. But we were doing it in the lab. We were finding that a calorie restriction and ketogenic could definitely manage inherited epileptic seizures. But we had a parallel study on brain cancer because we were looking at gangliosides in the brain tumors, trying to figure out what these complex glycolipids were doing. And then, when you have two parallel projects going on at the same time, you get some cross-fertilization. So we were looking at the role of gangliosides in blocking angiogenesis, which is an abnormal vascularization of cancer. In fact, it was the big thing around the turn of the century with Judah Volkmann and many others saying that you could, and even Jim Watson of the Watson and Crick fame for DNA, said that you can cure cancer by targeting the blood vessels. So we were looking at gangliosides and trying to block blood vessels by certain gangliosides definitely had an effect. But at the same time, we were doing calorie restriction for the epilepsy. And I said, why don't we put some of these, well, we had a drug. Listen to this. Some guy gave us this drug.
In a back parking lot somewhere?
Yeah. This guy They gave us a drug to block ganglioside biosynthesis, and I gave it to the mice that had the brain cancer, and all of a sudden the tumors were shrinking like crazy. And we said, oh, they were all excited. They started throwing money at our project and saying, oh my God, you have a drug a ganglioside targeting drug that can stop cancer by targeting the blood vessels. But then it turned out the strangest thing was that, this drug was amazing because the mice kept eating and they were losing body weight and the tumors were shrinking. And I said, wow, what's going on with this drug? So we put a body weight control in there, just mice that were given the regular food, but restricted to match the body weight of the guys getting the drug. And it turned out the tumor shrunk to the exact same stuff. Both mice had a shrunken tumor. One guy eating the drug and losing body weight, the other guy not taking the drug, but restricted in calories to match that of the guy taking the drug. And they both had restricted angiogenesis and shrunken tumors. And I said, wow, this is unbelievable. So
it wasn't the drug at all.
No, guess what? Well, what the drug did, listen to this. The drug blocked sucrases in the stomach, which means the food that they ate, they couldn't digest. So it was an indirect kind of calorie restriction. Right. So when I did all the control experiments, and I told the company that this whole drug was causing a failure to metabolize the food in the gut. And they were getting an indirect calorie restriction. Well, they didn't want to hear that at all.
Yeah, even though it worked because of that.
Yeah. They wanted to know they had a special drug to reduce angiogenesis in cancer, but it turned out to be calorie restriction. So indirect and they said, well, forget about that. We're not interested in that. And that was the end of that interaction. But then it brought us to the idea of what the hell is going on. So that's when I found Otto Warburg's work from the giant German physicist. He was Germany's most famous biochemist. Fascinating story about Otto Warburg, half Jewish guy, why Hitler didn't put him in the chambers. It was very interesting. Hitler feared cancer. He knew Warburg was the guy on the planet that would cure cancer. So Warburg was allowed to continue his work. And what he said was that all cancers are driven by glucose and a fermentation metabolism.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So when we started doing calorie restriction in the mice, I noticed that the blood sugar would go down and the ketones would go up and the tumors would shrink. It was anti-inflammatory, anti-angiogenic, and it killed the tumor cells. So I was saying, I'm seeing the exact same thing Warburg saw. So, and now we're starting to reevaluate this whole cancer thing in light of what Warburg did. So I did a deep dive on Warburg and found out, and I had German translations of his work, and I read his papers over and over again. I had the students read them over and over again. And we began to see the connections that this was a metabolic problem. And then at the same time, being a geneticist, I realized that the cancer cells were forming a reactive oxygen species, ROS, which would indirectly cause mutations. And then there were some beautiful studies that were done where you could remove the nucleus of the tumor cell and put it into the cytoplasm of a normal cell, and you got suppression of growth. And then you could take the nucleus of the normal cell and put it in a tumor cytoplasm, and you got dysregulated growth. So this was telling me that it was a cytoplasmic phenomenon, cancer, dysregulated growth. It wasn't due to mutations in the nucleus. So I put a big paper together saying cancer is a mitochondrial metabolic disease, which was met with some resistance at first, but now it's becoming more and more recognized.
Can I ask you a question with that? So obviously we have a lot of patients who are going to say, well, what is mitochondria? How is it important? What does it do for the body? Can you explain a little bit of that, please?
Yeah, I think this is a really important point. Mainly for those of us who have been in biology and biochemistry, we all know what the mitochondria is. But most of it's what you see. When you look at a cell under the microscope, the first thing you see is a big nucleus. I mean, this is the first thing you see. And there are chromosomes in there and the DNA and all this, and people have been following in the inheritance of genes, and it's in the nucleus. That the mitochondria is in the cytoplasm, and the mitochondrion is, you'd be surprised, it's really quite, if you look at drawings from textbooks, it shows this little bean-shaped thing in the cytoplasm. They call that the mitochondria. That's so unrepresentative of what this organelle really looks like. It's a spaghetti kind of network all through the cytoplasm. They fuse together, they separate, fission and fusion, all related to the biology and biochemistry of the cell. It's a dynamic organelle, unbelievable. That organelle determines the health and vitality, the metabolic homeostasis of the cell and the body. It determines when a cell should remain what we call differentiated, quiescent. It determines if a cell needs to divide. It determines how many divisions it should have and then stops. It determines if the cell is injured in some way. It has a self-destructing, what we call apoptosis, programmed cell death. It controls the destiny of the cell. That organelle controls the destiny of our very bodies. How long we live on the planet is
so not the DNA, but the substance around it.
Yeah, it's the energy. It's the energy. It controls the energy of the cell. And don't forget, it derived in evolutionary terms. It was a bacteria that entered into another cell. It was able to capture the oxygen and produce. That's how we develop metazoans, which are multicellular organisms. Before oxygen entered into the atmosphere 2.5 billion years ago, there was no oxygen in the atmosphere, but we had these cells that were there, single cells, and we have fossil records of these single cells, and they were all, they would have no regulation of proliferation. They called it unbridled proliferation. The only thing that would stop them is when fermentable fuels dissipated in the microenvironment, and then these cells would die. So then when this fusion happened with a proteoarchaeobacteria, you then were able to do division of labor. This was an organelle now that could produce energy far more efficiently than just fermentation. So in our normal cells of our body, as we speak and sit here, we're breathing, and the oxygen coming into our lungs is distributed to these cells, and the mitochondria in the cells are producing energy in a highly efficient way, extremely highly efficient. And if we were to stop breathing for a short period of time, the body would switch to a fermentation metabolism, and you would start to see lactic acid and succinic acid build up in the bloodstream. This is like a fail-safe to keep our cells functional for a short period of time while we're not breathing. Like if we were to have a heart attack patients stop breathing because the heart seizes and the bloodstream fills up with lactic acid and succinic acid. And if they don't get oxygen or start bringing that heart back, you're dead, because your neurons in the brain are going to die. They can't survive for very long without oxygen, nor can cardiac myocytes in the heart themselves. So if you keep that oxygen off, these cells up and die, and you're dead. Okay, but as soon as you start breathing again, the lactic acid and the succinic acid in the bloodstream disappear. The lactic acid and succinic acid are what we call waste products of fermentation metabolism, energy without oxygen. And before oxygen came into the atmosphere 2.5 billion years ago, all of the cells at that time were fermenters. They had no oxygen. And the interesting thing now is that cancer cells, even in the presence of 100% oxygen, continue to throw out lactic acid and succinic acid, just as if that's because their mitochondria are dysfunctional and they have fallen back on these ancient fermentation pathways. Which leads to dysregulated growth because, as I said, the healthy mitochondria determines how many cell divisions. It concerns the differentiate. It concerns quiescence. It controls our very existence. That organelle is so powerful and so necessary for normal biological function that if something happens and the mitochondria causes what we call apoptosis, immediate cell death, then the cell will die. But when that organelle becomes corrupted from things in the environment, and I can talk about all the things that initiate cancer, the cell gradually shifts to this ancient fermentation pathway, no longer regulated, because the organelle regulating the destiny of the cell has been corrupted, and it falls back on the ancient pathways 2.5 billion years ago. So we put the whole path of this cancer thing into a new perspective, and we know these tumor cells cannot survive without fermentable fuels, and the two fermentable fuels are glucose and glutamine. And
let's go an easy
I know I put a lot of stuff out there.
That's all right. You're saying that the conventional wisdom is cancer is a DNA problem. Your DNA is allowing your cells to reproduce too fast. You're saying it has nothing to do with the DNA. It has to do with what's going on with the mitochondria, and it's not stopping the cell division.
Yeah, I mean, we know that the mutations are downstream effects of the damage to the oxidative phosphorylation in the mitochondria.
That's what I was going to ask you about when you talk about the oxidation phosphorylation in the production of ATP. You know, there's the certain supplements and things out there that people can, that we can take and recommend to our clients and patients. And that's one of the things we wanted to ask about too was
how do we optimize the mitochondria?
Yeah, and you're hearing about the benefits of creatine and methylene blue and NAD and ivermectin and all these different treatments.
Well, you have to remember one thing. You've got to separate the biology of the normal cells in our body, because not every cell in our body has cancer. You're talking about a cancer and a tumor. Those cells have a different biochemistry than the normal cells of our body. So what our strategy is, our press pulse therapeutic strategy, which we developed based on our understanding of what cancer is biochemically, we have to take away the two fuels driving the dysregulated growth in the tumor cells, but we have to do that without harming the normal cells in our body. So we know that glucose, the sugar glucose, is a prime driver of dysregulated cell growth in the tumor cell. But we also know the tumor cells cannot burn fatty acids or ketone bodies, because you need a good, healthy mitochondria to use fatty acids and ketone bodies for energy. So by lowering blood sugar and increasing ketone body, this is what happens when we stop eating. This is our evolutionary history. During the Paleolithic period of our existence as a species, our ancestors were mostly in nutritional ketosis, mainly because they didn't have access to high carbohydrate, highly processed carbs.
They had to hunt and gather, right? Yeah.
Yeah, they were hunting and killing animals, and it takes a lot of energy to spear a buffalo. Or a woolly mammoth. I mean, figure it out, man. We don't have a gun. We got spears and rocks.
Right.
Then we have to use sharp tools to cut off the skin. I mean, you're talking a lot.
To earn your food.
Here. Yeah. And then you're going to chow down, marrow in the bone, all full of fat. And we did not have delicatessens and donut shops on every corner during the Paleolithic
and you weren't
period.
cooking them with seed oils either. So
none of that.
Right.
So people always say, well, those people didn't live very long. Well, let me tell you, those people died from injuries and infections because they didn't have an orthopedic surgeon to help them with the broken bone, nor did they have antibiotics.
Right.
So they
would live forever in today's society.
Oh yeah.
They died healthy, basically.
Yes, absolutely. Yeah. But but that was our ancestry, and when you go back and live that, like my friend Dominic D'Agostino, he's always in one of these ketotic states. He's like a Paleolithic man in modern times. His ketones are always at 1 millimolar, maybe a little higher, and his blood sugar is always down. So he's in a state of nutritional ketosis. Well, when you take a cancer patient and put them in that state of nutritional ketosis, Their normal cells are getting healthier and healthier by burning fatty acids and ketones, but the tumor cells are restricted. They can't burn that. So they're dependent on glucose. And our big discovery that Warburg did not know about is we were able to show that the amino acid glutamine, which is the most abundant amino acid in the human body, in the blood, is also fermented, not respired. So these tumor cells as long as they have access to high levels of glucose and glutamine, they grow like crazy and then you try to kill them with poisonous chemicals and radiation, and the next thing you're dead from the treatments.
Yeah. Well, that's
the cancer.
That was one of the questions I had when you talked about that's the one thing that we never addressed or entertained with my dad's cancer journey is the amino acid glutamine. And I've been a natural bodybuilder my whole life, and we were always supplementing glutamine and it's the most abundant amino acid in the human body, responsible for your white blood cell count, so important. But yet I hear, and I've learned all this from you, from watching your videos, how do we address the balance and the importance of glutamine that it plays in the body, but also the fact that if you have cancer, it's feeding cancer.
Yeah, well, that's the important part. This is why we developed Press Pulse. Pulsing is the drugs that use to target glutamine. So because there is no diet that we can use to reduce glutamine. And we just published a paper showing if you have super high levels of glutamine, you can force the tumors to grow faster. But already, you see, the thing of it is, the cancer cell doesn't need, it needs glutamine, but the amount that we already have in our bloodstream is necessary and sufficient to provide enough glutamine for the tumor. So that's why we have to use glutamine targeting drugs. But just like you said, these drugs have off-target effects, so you have to be very careful. Because if you target glutamine too aggressively, you actually disrupt the immune system, which also needs glutamine, and your microbiome, the gut. So the gut uses tremendous glutamine, and your immune cells that are needed to pick up dead cancer cells also need the same thing. So the cancer cell and the immune cell both use the same metabolite. So you have to put a chokehold on the glucose first, which puts tremendous pressure on the cancer cell. And then you have to come in and strategically use the pulsing of glutamine targeting drugs so that you don't damage your gut or paralyze your immune system. And that was
glucose alone, if you stop the glucose alone, does that get rid of the cancer or just slow it?
It usually slows it down. There are some cases where it just completely dissolves the cancer. We're shocked about that without even targeting glutamine. But I think that the best way to do the management of the cancer is to it's easy to get glucose down because we have so many ways to do that. Water-only fasting, exercise, a lot of ways to get your glucose down, put yourself into nutritional ketosis. But then you come in and strategically hammer out the glutamine. So you degrade the tumor slow. Because our plan is that if you kill a lot of tumor cells, you have these dead cells in your body, these in and around the tumor. And what removes them is they got to get rid of the corpses, and that's the role of macrophages and some of the immune cells. You got to come in and pick up the dead so you don't get infections and things like that. So you have to kill and then come in and remove corpses. Kill, remove corpses. Kill, remove corpses. So it's a strategically lowering while the rest of the body gets healthier and healthier and healthier, burning ketones just like our Paleolithic ancestors did. You're burning ketones, making your muscles and the rest of your body super healthy, but you're slowly degrading the tumor, knowing that its biology is dependent on glucose and glutamine.
What I'd like to do, if I could machine gun a few questions regarding some of the treatments that I'm using currently in our clinic, of which I'd like to connect with you. I understand you guys are kind of looking at other places to work with who are treating people in the unconventional manners. We can talk about that afterwards. But some of the questions I'd like to do, if we could, is like, number one, we talked about fasting and pulse fasting, I'm assuming as well, because you don't want someone to be obviously nutritionally deficient. So can you quickly speak to the benefits of fasting and how long and a little bit of the details regarding that?
Yeah, so one of the things we developed here at Boston College was the glucose ketone index calculator, the GKI. Okay, so the GKI lowers, it measures the ratio of in millimolar in glucose and ketone in the bloodstream. So when you do water-only fasting, your GKI goes down. And it depends how low your blood sugar goes. I mean, if you're in 65 to 75 milligram per deciliter, which comes out to about 3 point something millimolar, and you can get your ketones up to about 2 millimolar or something like that, you can get down into these low GKI values. And you can do that with water-only fasting, or you can do it with various diets like carnivore diets and things like this. That don't have to be as harsh as water-only fasting. When we first did this, we were saying that you could dissolve cancer by just doing water-only fasting, and then oh yeah, well that's good. Let me try that. Well, how about doing it for 30 days? Well, you guys must be nuts. Well, who's going to fast for 30 days? But I'll tell you, the guys who do fast for 30 days, the body turns on the tumor and actually uses it for food. It's called autolytic cannibalism. Oh yeah, the body will look at when you don't eat for that long. And you're healthy. Now you can't be, you have to be, you have to be careful. You have to be careful in doing that. But a lot of people who have cancer have type 2 diabetes, hypertension, high blood pressure. They got all kinds of other comorbidities. You can't take a person like that and say, don't eat for 30 days. But what happens is that we generally take, I don't, because the physicians and PAs and people that I work with, we generally take these people with cancer, and we have to do a complete blood work on them at first. And you find out how many parameters of the entire panels of everything we can measure, vitamins and lipid, everything. And we look at parasites in the blood. You can't believe how many people have parasites. You have to take that person, slowly move them into a better nutritional state. And you can use what we do is zero carb diets, generally for about 10 days. Which it's easier to do that when you do a carnivore with a zero carb diet, and also restrict it, not like eat all you can eat, and then gradually work them into water only fasting. And then when they get to these zones where the glucose ketone index is 2.0 or below, then you come in with the glutamine targeting drugs. And then you can polish off. These cancer cells are now super vulnerable to death by just tweaking a little bit. And we don't rule out chemo or even radiation for most cancers. It turns out that you could use extremely low doses of chemo and radiation, and these tumor cells just up and die like you can't believe.
Because they've been weakened by the diet.
Oh yeah, they're
Instead of coming in and just blasting somebody from day one with a high dose of chemo and then just
that's absurd.
Cooking on the organs.
You don't do that. If you understand the biology and biochemistry of the disease, you never do that. You would never do that. But you can use those same chemo at extremely low doses. Yeah, we learned that from our clinics in Istanbul. When you put these people in nutritional ketosis for a couple 3 weeks, and then you come in with low doses of chemo, they're extremely well tolerated. The patients no longer have this terrible sickness and all the kinds of adverse effects. And you can even use a little radiation because the body now, the normal cells in the zone are super healthy and resist damage from radiation and chemo, whereas the tumors are super sensitive to radiation and chemo. So
you mentioned that because of the lactic acid buildup and the side effects of having cancer, that chemotherapy and radiation don't work because of the acidotic state. And then once you start improving this situation and they become, I guess, less acidotic, then therefore the chemos work better. Yeah. But that did raise a question regarding the ketosis acidosis state that you get when you are in ketosis versus a lactic acid buildup. Thoughts on that?
Yeah, well, that's usually for type 1 diabetics. Ketoacidosis will be 15 to 20 millimolar of ketones in the bloodstream. And not only that, you have high glucose, keto. This is a deadly.
Good point.
You're talking about nutritional therapeutic ketosis. You're down in the range of 3 to 4 millimolar. You're not even close to what ketoacidosis situation is. You can at 6 to 9 millimolar ketone in the blood, you can sometimes get heart palpitations at that zone, at that level. But most of the people who we're killing cancer with, they're in the 3 to 5 millimolar range. Occasionally they'll go higher. It depends on how your body handles it. Obviously, if you start to feel palpitations, you might want to take tiny amount of carb that will reduce it. Again, every physician that works with a cancer patient should know the parameters of what's going on. So we're developing an app that will measure glucose, the GKI, to allow the physician and the patient to know what zones they're in, whereby they can then apply different drugs and procedures to kind of polish off the cancer over time.
So look, one question I had about that. So, we talk about the low carb and going into keto, the gluconeogenesis, where your body's going to turn, fatty acids and proteins into glucose for the, and so that would be the argument that I've had with some of the traditional doctors and things like that. So, well, your body's going to have glucose in it regardless, even, because of homeostasis. So how does that part play when you're not feeding the initial source of carbohydrates in your body for that initial source of fuel,
then
and it's going through homeostasis?
Okay, then we have to look at who's using the sugar that's being. Don't forget it takes energy to make energy through gluconeogenesis. Right. Yeah, you'll target amino acids and things like this, but the organ that's going to get most of the glucose is going to be the brain and the muscle.
Okay.
So not the tumor cell. So you're talking about a much lower level of glucose now circulating in the blood, right?
Exactly.
So those tumors. So you just go out and walk 4 miles a day. And your muscles are sucking the little blood made from gluconeogenesis, and the brain's taking the rest. There ain't much left for these poor tumor cells.
Well, that's the answer, and I didn't know the exact answer, but that totally makes sense. And that was the argument that people would have. Well, you can't get rid of glucose and glycogen in your body.
It's all GKI.
And that's the importance of exercise then too. Yeah. And pushing exercise is one of the treatment options.
Absolutely. You can and it depends on the exercise and how much you do and what you do. So you can do weightlifting, but it's not to do it to the point where you're totally exhausted.
Right. Resistance training is going to be great for, burning up glucose or glycogen in the muscle. Yeah. So, you're going to help with depleting out that first source of fuel.
Yes. And as one thing you have to realize, the muscle does not share. So, and that's an evolutionarily conserved adaptation, because if the muscle were sharing its sugar with other organs and the bear comes out of the woods and you have no longer ability to run, you're dead.
Right. Yeah. Good point.
So the muscle keeps the sugar. So you exercise and burn sugar, the muscle's going to be sucking it out of the blood and not sharing it with the tumor cells. I can tell you.
And you don't want to stress exercise so much that you produce cortisol because of the stress on the body. So
well, this is another thing. We use stress management, emotional stress. See, when people have cancer, they get all freaked out. They think they're going to die. Oh, the angel of death is looking over my shoulder. We use a lot of ways to reduce cortisol. You have other stress management techniques together with exercise. And also, the confidence of the patient builds up the more and more they keep the pressure on these tumor cells, because you do a non-invasive analysis of the tumor, whether it's CAT scan, MRI, PET scan, and you come back and you see, or even the size of the lump or whatever it was starting to shrink, and you start to see evidence that this thing is getting smaller. That makes the patient feel stronger and healthier. All of this comes into play because the energy that you're expending to manage this is falling now a lot on your shoulders, and you're not just sitting there as some sheep being treated by some person who doesn't really understand the whole problem. So the patient with metabolic therapy becomes a real participant in the overall resolution of the problem.
So just to summarize, you're saying instead of thinking of cancer as the angel of death, like you were just saying, it's more like, okay, I've got cancer, so I have to get into a different food source. I have to get rid of the carbohydrates, live off of ketosis. The second thing I need to do is a little bit of exercise to get rid of what sugar there is laying around. And the third thing is I have to get a better state of mind. None of those require drugs. None of those require going into the hospital.
Well, this is the most but this is the biggest resistance to what we're talking about. Yeah. The fact that a lot of this doesn't require drugs. And that's a stick in the spoke to an entire industry. So the resistance that we see for cancer as a metabolic disease comes from, the resistance of the whole system.
The profit centers.
And I'm sorry to say this, cancer is a revenue generating disease, a massive revenue. You're talking about managing a disease based on evolutionary concepts, dietary changes, exercise, and we're not ruling out drugs. Listen, we have drugs that we need to facilitate this, and I don't see it to be such a undermining. If you're taking a certain amount of cancer drug like immunotherapy. Let me get back one thing about the immunotherapies, because you hear them advertised every night on the TV about Opdivo and Keytruda and all these kinds of things. It's called immunotherapies. They're based on the cancer being a genetic disease. But they say, oh, these immunotherapies don't work because of the acidification, which you mentioned previously. Well, when you pull the plug on the glucose and glutamine, you remove the two acidifying molecules, lactic acid and succinic acid, now potentially making these immunotherapies work much better. But you can use them with lower dosages. Same with doxorubicin, which is called Red Devil death. These drugs are so toxic to your body, but you can use them, but you use them in different concentrations. So that means you can still use these very expensive drugs at half the dosages, but still charge the same amount. So it doesn't really hammer your bottom line. But my goal is our goal is to see how long we can keep stage 4 cancer patients, or any cancer patient, that way. And we never use the term cure. When we say we use metabolic therapy, we use metabolic therapy to manage cancer. Now, if you're supposed to be dead in 1 year and you're living 5 or 6 years with a higher quality of life and overall survival, that's success. I don't care what anybody says. That's success. Whether that person gets cured or not, we don't know. All we know is we're not harming the body of that person. We're making them stronger.
Yes.
Right. And if they are cured, so that's the better part. But I never use the term cure. I told my students, never. It's an arrogant term, making it seem like you can know something that cannot be known. So
adding to the next step of treatment options, obviously supplements, hyperbaric chamber. Some of the supplements that I've researched, and I'm sure you've come across them as well, berberine, turmeric, green leaf or green tea extract, vitamin C, and even probiotics. Can you speak to those and then add a little bit about hyperbaric chamber?
Yeah, well, those are all okay. But if you're not in a low GKI, they're not going to have much effect.
Right.
Some of it, you get a lot of effects from placebo effect. It makes you think you're doing, just by taking some of that stuff, you feel like you're getting better. Placebo can be very powerful, a very powerful effect. But we don't discourage berberine. None of those things. They'll have small effects. They're like little pushes. They can help the system as long as you can keep that low GKI and take berberine. What we're finding is with only parasite medication that we know of that we've tested ourselves is embendazole. It's like fenbendazole. And the reason why the parasite, the reason why I didn't try ivermectin, we were going to do that a couple 3 years ago, but the politics around it was too. So we decided not to do ivermectin. But
so we call him the ivermectin guy around here because he got really popular during COVID, putting his patients on the ivermectin and a lot of success.
That's the reason we didn't touch it, because of the COVID crazy stuff going on.
Because it worked too well.
Yeah, well, it might have, but you see, in cancer, when you use a drug, you have to have knowledge about the mechanism of action. Why is it killing tumor cells? So when I was doing research, it was really interesting because parasites use mitochondrial substrate level phosphorylation, which is what we've discovered is the new thing in cancer that Otto Warburg did not know about. Was that there is that glutamine is fermented in the mitochondria itself. This is I'm getting now deeper into the science, but it turns out that the damn parasites use the same pathway. They ferment glutamine. So I said, if the goddamn parasites can be killed by these drugs using the same pathway that the tumor cell uses, it should kill tumor cells as well. And when you choke hold the glucose and then put in the embendazole, which targets the fermentation in the mitochondria, you start slaughtering these tumor cells. Only because the parasite and the cancer use the same metabolic pathway. So that's the mechanism. And also, embendazole inhibits glucose uptake. So you have a drug that does two things. and works so much better under nutritional ketosis. So we're able to understand the metabolic parameters and utilize that for killing tumor cells.
What
there
was
a
your
difference-
what was your therapeutic dose milligram per kilogram that you found to be effective?
Well, this again is dependent on the gender, the body weight, all these different kinds
Right.
of things. We started with what they used for killing parasites, which was 200 milligrams a day for 3 days on, and that usually slaughters a lot of these parasites in the body. And we got a lot of this knowledge from China and India, where they have parasite infections, and those guys were doing research on this for decades. So we started to adapt. As a matter of fact, the big paper we published with my colleague Thomas Durai, who's an MD, Ph.D. in my laboratory, he has in that paper he talks about dosages, timing, and all that for people that would have this, and it can vary depending. The physician and the patient have to work together. How do you feel? That's the bottom line. How do you feel? Oh, I feel good. If you take too much of it too often, you can harm your liver. Everything has to be done with a interaction between the patient and the physician to know what levels of dosages that are working best. By looking at number one, how the patient feels, and you use non-invasive technology to see how the tumor is responding. And you can use blood work. So there's a lot of ways. So it's a very close interaction between the patient and the oncologist, applying all these different strategies, exercise, putting them all together, looking at your GKI. How do you feel? What does the tumor look like now? Is it shrinking? All of these kinds of things. And then the patient must be collecting data on themselves. And this is another thing that we need to publish these papers. We have to have the participation of the patient. This is why our Pablo Kelly, the person who lived 10 years with a glioblastoma, was collecting data. I didn't want to publish his paper, but he had 5 years of constant measuring his glucose ketone index, 5 years. So we're able to put a paper together to show how it works. He was never cured, but the tumor was managed through periodic debulking.
And he was the gentleman who died from the cerebral hemorrhage from surgery, correct?
Yes, we spoke to him, but he was happy-go-lucky. I talked to him for years. Nice guy, very nice guy out of Devon, England. But he wanted that last surgery, thinking he would get the final residual piece of tumor. But unfortunately, 12 hours after he had the surgery, he had a cerebral hemorrhage and died. But let's be honest, the guy lived for 10 years.
Right. And he was able to have 2 kids, thanks to you guys.
Oh, yeah. And he never really got serious on targeting the glutamine. So he was pretty much hands. He didn't want any drugs. He was one of these holistic kind of guys. We're going to do it by all natural stuff. But we could have kept Pablo alive had he not. I think we could have. He would have still be with us if he didn't have that last surgery. But we're seeing more and more success stories like this over and over again. We're trying to publish these papers. I need to work with clinicians, and we need to work with patients. So and we write it up here at Boston. It's not easy writing case reports because you're looking at the data, and all of a sudden the guy's got a GKI up at like 65 or 70 when he was down at 2. I said, "What the hell?" I decided to have a pizza and some Coke. Well, our mice don't do that. We have that.
Right.
So my colleagues in the lab say if we could take these cancer patients and put them in the cages like we do the mice, we would have them taken care of real quick. But
that goes back to like one of the nutrition questions I had. I was wondering about what's we would say the law of diminishing return between lowering your glucose, and there's you have foods like blueberries and things like that have the phytonutrients and the micronutrients that help kill cancer, but yet it's a carbohydrate source.
Well, but you don't need much, because those phytochemicals, a lot of those things, you don't need much of that stuff to be really therapeutic.
So you could supplement it, yeah.
Yeah, and you could test once you're in that low GKI, people will experiment on what they can or cannot eat. So, and vegans, if they want to try it, my hat's off to them. It's hard to do that on a vegan diet, but carnivore is easy. Pescatarian, eating fish, And our colleagues in Greece just published a big paper. I was one of the authors on the paper for glioblastoma, where they used a calorie-restricted Mediterranean diet, sardines, salmon, olive oil, avocados. Not the worst thing in the world. You can eat. But what about you eat tomahawk ribeye? I mean, that's a lot of people would like to eat that. You just don't eat much of it. It doesn't raise your GKI. So people always say, "Oh, these ketogenic diets are horrible." Well, it's not a ketogenic diet. It's nutritional ketosis. What keeps you in the zone of nutritional ketosis?
So if you combine the keeping your body in ketosis along with keeping a high oxygen state in your body, he mentioned talking about exercise. I got a hyperbaric chamber that I put in my basement, and for my dad for his cancer, and of course I use it myself for health and fitness and bodybuilding. But do you have much experience with the hyperbaric HBOT therapy?
Yeah, we did some research on preclinical studies with my colleague Dominic D'Agostino from the University of South Florida. He's the big, he works with the Navy SEALs. He works with all these things in hyperbaric chambers and all this kind of stuff. He was the one who pointed out to me that these cancer cells under hyperbaric treatment exploded and blew up. And what's happening,
Meaning went away or got worse?
No, they were killed. They died.
They killed. Yeah,
Hyperbaric
killed them.
Killed them.
Wow.
And we're finding that when patients are in nutritional ketosis, whatever time of day that happens to be, when your blood sugar is low and your ketones are elevated, hyperbaric oxygen will create tremendous oxidative stress in the tumor cell. So, and the ketones protect your normal cells against any oxidative stress. So the low glucose creates oxidative stress in the tumor, and then you put them into a hyperbaric chamber at about 2.5 atmospheres for an hour. And you'd repeatedly do this. This is another add-on to do that. For brain cancer, you have to be a little careful. It's always important to be in nutritional ketosis because you don't want strokes and things like that, which can happen if you're not in nutritional ketosis. So, but for other types of cancers, lung and lung, you have to be careful about too, mainly because of the oxidative stress on the tissue. But other cancers are usually quite readily managed. What you call IV vitamin C will also create oxidative stress on the tumor, also working better when the patient is in nutritional ketosis.
The real high dose vitamin C, like get the hydration bag and get the real high dose vitamin C.
You have to have it IV.
Yes.
It has to be intravenous vitamin C.
Yeah.
That
Excellent.
creates so hyperbaric oxygen and IV vitamin C kill tumor cells by oxidative stress. So especially if you're in a low GKI.
What would be your? I mean, I know you can go extremely high dose of vitamin C. What have you seen?
I see people do this and they claim they have this success. All we know is that we're always looking at the biology and biochemistry of the tumor cell under these different conditions, knowing what the stress level is to kill them. So you can use radiation kills tumor cells by oxidative stress, but you can use high dose vitamin C.
Vitamin C.
So you're killing the tumor by a similar mechanism, but you can use different strategies to do that. The key is how you kill tumor cells without harming the rest of the cells in your body. And that's what ketogenic metabolic therapy strategy is, killing tumor cells while enhancing the health and vitality of your normal cells. So when you have cancer and you have diabetes and high blood pressure, hypertension, you have all of the things, you do metabolic therapy, besides you have cancer, you do metabolic therapy and you come out of the treatment healthier than you did when you went in. So you got to get these people up front. You can't. The problem is sometimes they come and say, "Oh, I failed radiation, chemo, immunotherapies. Now can I use ketogenic metabolic therapy?" Well, your body's beat to hell. Your normal cells are all beat to hell. It's
so you're better off getting your body strong and doing the ketosis plan and things like that, and getting your body, and then introducing that small dose of chemo if you had to.
Yes, exactly.
Yes. Absolutely. And then you come out of it much healthier and your tumors are hammered. But we're not doing that in the clinics. We're not doing that. We're irradiating and poisoning people and surgically mutilating them. And we don't, I mean, and they've said, oh, Seyfried says the standards of care are medieval. Well, for crying out loud, they are medieval. Well, you
Right.
might just do another human being if you knew, I mean, for crying
So
out loud.
if he had a patient or a client of mine, or like something like my father, like what would be to work with you or your clinic and as a team effort between Keith and your clinic? Well, how would something like that? What would something like that look like? The people that we work with.
I get you hooked up with the folks that are in the clinic doing this or working with cancer patients, and they make recommendations. They can do it by Zoom. I don't treat the cancer patients myself. We do all the preclinical studies, which are with mice that have natural forms of cancer. And then we work out details, and then I share that with my clinical colleagues, and they work out differences in doses, timing, and scheduling based on what we do. Because, they like to know that what we're doing is not going to harm their patients. They already have cancer. Now you're going to do something to make them worse than they are. We want to make sure that never happens. We only want success. So Alicia Halakis out in Minneapolis has a clinic where she's working on cancer patients, and we're working with different clinics around the world, actually, in India, also developing the strategies for doing this. So again, it's not standardized. This is the problem right now. So every knowledgeable physician or caregiver has to have as much knowledge about what I'm saying, and then they would work with the patient together as a team, and then see how it works. And it doesn't work on everybody. You have to realize that it doesn't work on everybody, but it works on most people.
And you briefly mentioned something early on in our conversation about, I guess it was in India, the success they're having with stage 4 pancreatic cancer.
That was in Turkey.
Okay.
Yeah. And I'm going to go over there. We're working. We want to get enough data on these people to write up case reports. And we're working.
What are they doing differently, or is there something different than the protocol that we've discussed?
Well, we're using. Well, this is the thing, because it's such a new way of doing things. You're finding out that heat therapy and some other kinds of approaches, while the patient is in nutritional ketosis, are having
sauna.
Yes, well, not so much sauna. Hot pads on top of tumors.
Okay.
Yeah, you can, but that's localized. It's really interesting. William Colley developed the Colley vaccine during the last century. He would inject cancer patients with live staph and strep, which
Wow.
are yeah. So it's funny because it's not funny. It's just interesting. The patients that would survive the fever from the staph and strep would oftentimes have complete resolution of their tumor. So high body temperature caused by fever destroyed the tumor and the body. As again, the cancer cells are weakened and they can't handle the high stress of the heat. So 5% of his patients would die from the fevers. So he was killing 5%, but it was an acute death. You see, you give the live staph and strep, and a majority of his patients that could survive the fever were cancer free after the event. But in today's society, about 50% of people being treated for cancer are dying from the treatments. The problem is they don't die acutely like you would if you did staph strep treatment.
What's fascinating, what you said about the heat therapy and direct hitting it directly with like heat pads and things like that, is I'm really into sauna therapy myself, and when you're doing sauna, you're manually giving yourself a fever.
Yeah,
you're manually giving your body a fever, and that just makes sense. And I wonder if is there any information about that? Or?
Yeah, well, there's fever from bacterial infections where you reach a massive immune system attack. It's like the whole immune system goes wild when you have a bacterial infection in your body. The T and B cells, the macrophage, everything is like rallying together to kill the invasion. And raising body temperature through the release of cytokines is one way. This is not the same as putting your body in a sauna, because that temperature is external from the heat outside rather than the heat generated inside the body from the immune system. But nevertheless, when you reach a certain temperature, the tumor cell membranes can't handle it, and they die in a localized way.
There could be some benefit there.
Yeah, there is some benefit there. We like to study a little bit more on that because it does seem to have some combination of things. But you see, here's what I'm relaying to you is that we have so many new ways to mix and match different ways to kill tumor cells without harming the normal cells of the body. And this is the new frontier. This is the new paradigm. Oncologists, once they stop thinking cancer is a genetic disease and are trained in medical schools to know how many nifty ways we can kill tumor cells without harming the rest of the body.
Will that ever happen?
Well, that has to happen if we really want to drop the death rate. Otherwise, we're going to continue the status quo, which right now is 1,700 people a day dying from cancer, which is about 70 an hour. And most of that is needless. That should never be happening in today's day and age.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paperbook via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just...
One thing that we really haven't discussed much is the glutamine blockers. And are there medications that can be used off-label currently that we have available to us to help with glutamine blocking?
Well, the Dawn drug, 6-deoxynorlucine, which was used for cancer in little children and in the past, was discontinued because it did have toxic effects on the gut and mucositis, I should say, in the gums, and like many cancer drugs. But embendazole targets the glutamine pathway. We have a paper showing that, but you have to be in nutritional ketosis to have it work maximally. It would be great if we could get that. We published a paper showing how when we used Dawn, we could use lower dosages and reduce the toxicity of that drug. I think we can reduce the toxicity of any of these drugs when the patient is in nutritional ketosis. And not only that, it facilitates delivery of the drug through the blood-brain barrier for the brain cancer patients.
Nice.
By a mechanism we have not yet ferreted out. But we know it is because we did electrospray mass spec to show how much more drug is on target with lower dosages of drug. I mean, this managing cancer should not be an insurmountable challenge. We now know what to do and how to do it. The problem is the system is not accepting this because they prefer to keep the status quo. It's very profitable.
Right. So with the status quo, you said yourself, it's like half the patients die from the treatment, not from the cancer. What can we do, or what kind of success have you seen with your techniques of, first of all, get into ketogenesis, second of all, then start targeting other things? What's the success rate with that versus chemo?
Well, we're probably starting to publish Pablo Kelly's paper. He lived 10 years. I mean, you don't really get GBM patients living 10 years under any conditions. Glioblastoma, most people, we haven't made any advance in 100 years in glioblastoma.
Wow.
In 1926, the survival for glioblastoma was between 8 and 14 months. And today it's 8 to 17 months. So, I mean, what are you talking about? No major advances in glioblastoma. That's because they irradiate the brain. And when you irradiate the brain, you free up massive amounts of glucose and glutamine in the microenvironment, the very fuels that are driving the dysregulated growth. And then you give high-dose steroids because the brain is swelling from the radiation, which brings sugar up level, rocketing sugar. And then they wonder why we're not making any progress. I mean, you got to stop irradiating the brain, man. And when you do that, you're going to get much greater survival. But even if you irradiate the brain and put patients into nutritional ketosis, our study just came out of Greece, where they live significantly longer, beyond 3 years, if you did the Mediterranean ketogenic diet than if you didn't. So clearly, we're showing great success. Getting back to your question, how of these stage 4 cancers, how long are we keeping people alive? And the answer is much longer than they would have if they were treated with standard of care alone. How
And
much?
they feel better.
Oh yeah, they feel a hell of a lot better. So and when they die, if they die very peacefully. Some of these folks have relayed information to me, and they said, he was really good up until the day before he died. He didn't suffer like being horrific long, but he lived 4 times longer than he would have. And when he died, it was peaceful. It wasn't this horrific wasting away from being poisoned and irradiated. So we see success. Now, again, this is all new, guys. This is all new. We're perfecting as we speak. We're developing more and more effective strategies. So
can I ask you this, Doctor? What would you say to somebody like the challenge that I have, like if you can't get somebody to have the discipline to do keto, right? And we're trying to get them to doing all the supplements or doing all the protocol, but they can't, like my father, what do you say to somebody there? Like, and is there any ideas or tips? Because
you have to ask why can't they do that? Why
do they choose not to, basically?
Or what is making them not want to do? Yeah. Well, it's not only discipline, because sometimes you're using things that are really that person doesn't like, or finds it inconvenient, or
they want to eat carbohydrates, they want to eat sugar, they want to eat the good tasting.
Well, then see, this is why we're developing the handheld app, so the patient can know right off the bat. We have zones on the app. You have zones that we call cancer driving zones, or cancer inhibition zones, prevention zones. How do you prevent cancer in the first place? How do you prevent all these diseases, chronic diseases, in the first place? You take your
blood sugar and then register in the app.
We're developing it from continuous glucose ketone monitors.
Okay. Yeah.
But right now you have to take a finger prick to know that.
And so you'll adapt it to like a Stella or whatever the different.
Yeah.
Okay.
Yeah. So you'll have real time like if you want to eat a candy bar and you're under cancer, and you say that I just ate something that's causing my tumor cells to start aggressively growing,
right?
And now you know that. Well, is that what you really want? Is that
right? Exactly. Yeah. It's, and I can't understand it, but there's some people who they won't.
But when you put it on a color-coded chart and it tells you exactly what that food or what you're doing is doing to the cancer cell, you can't see it happening, but you can feel it happening. Right. You start changing your behavior. Don't forget, our Paleolithic ancestors were in that state, not because they wanted to be, but that's because they had to be.
No choice.
There was no choices.
Right.
And the problem today is we have these choices, which then challenge us to be in these zones.
And the non-compliance really messes with the data when we're trying to heal people and they're being non-compliant. It's making us look like our therapies aren't working. And then they go rely on the Western medicine. They tap out, they get scared, and they go into the chemo radiation world and then end up dying. But they never really gave themselves a fighting chance because they weren't disciplined or they had the psychological blocks. And that's why I like that you mentioned stress management being a piece of this, is that if you can get them in the right headspace, then you can get their bodies in gear. Right.
Well, but that's the problem right now, because many cancer patients go to their oncologists at the top medical schools and are told that diet and nutrition have nothing to do. Glucose has nothing to do with any of this. Eat whatever you want.
They told me it's bad luck. That's what they told me. So
that's based on the somatic mutation theory, which is now debunked. We went out of our way. I mean, we have so much evidence to say cancer is not a genetic disease.
Right.
You have some tumors that have no mutations. How you explain that? And then you have all these so-called driver gene mutations that were supposed to be responsible for dysregulated cell growth. They're present in our bodies right now, mutations in driver genes, and we don't have cancer. And then you have the nuclear mitochondrial transfer experiments that are really showing that cancer cannot be a genetic disease. So
Wow.
why are you building therapies? Well, radiation and toxic chemo, they don't really care whether it's a metabolic disease or a genetic disease. But immunotherapies like Keytruda, Opdivo, and CAR T immunotherapy and all these kinds of things you hear about, that's based on the somatic mutation theory. And if the theory is incorrect, those approaches will never be optimal. Every now and then you get somebody who really responds well, but the majority of people eventually don't respond, and many of them are killed by the off-target effects of some of these immunotherapies. But we can still use them if we can remove the acidification of the microenvironment. Then they might work, and that's when you take the glucose and glutamine away, and now the tumor cells could be extremely vulnerable not only to immunotherapies but to many of the other standards of care that we're currently using them at lower dosages so they reduce the harm to the rest of the body.
That reminded me of a question I had on here was about cancer stem cells. So we've done some labs on my dad testing for cancer stem cells because,
Molecular residual disease.
because the toxic treatments can't even touch the stem cells anyways. But I've seen some things on ivermectin and the parasite, drugs, the indirect, using it to starve the stem cell itself.
Yeah, well, that field is all excited about cancer stem cells. We have cancers in our lab that are both stem cell origin and mesenchymal origin. Now, you have to realize that all the metastatic cancers are of mesenchymal origin. They're macrophages, where stem cells can't spread. Stem cells will grow like crazy in situ in a localized area, but they can't spread around your body. They don't have the genetic blueprint to enter your bloodstream, survive the immune system, and then set up sites local. All of those cells are massively dependent on glutamine because
Stem cells?
Yeah, no, not the stem cells. Stem cells can be killed easy because they just require the glucose. They can't spread, so you can focally kill them. And they're still all our stem cells are glucose and glutamine dependent as well. They're just not capable. They don't have the genetic capability of spreading because they're not macrophages.
Isn't this why we get so many cancer reoccurrences? Somebody will get, they get diagnosed with cancer, they get chemo treatment for 6 months a year, and all their tumors gone down, everything looks good, and all of a sudden they get a reoccurrence because they can't tap into the stem cells.
Yeah, well, that could be, but we're equal opportunity killers of cancer cells. We know what they need to survive, whether they're a stem cell or a metastatic cell, and we know how metastasis happens because we know there's fusion hybridization. So you have a tumor that's full of stem cells, and it elicits a response on the part of the body like a wound, as if you had a wound in your body somewhere. So your immune cells come in to heal the wound, and they throw out growth factors and cytokines, which are the normal strategic activity of the immune system. And what happens, which actually makes the stem cells grow a little faster, and then it's like an escalating situation of biological chaos. The very immune cells that are supposed to be putting out the fire begin to fuse with the stem cells, with each other and the stem cells, and then you create a rogue macrophage, a cell that already is capable of entering and exiting tissues, except now it's mitochondria defective, but yet it becomes dysregulated and spreads all over your body. And that was from a fusion hybridization between the immune system and the stem cell. So, but the stem cell by itself can't do that. So it has to have this fusion hybridization. And they're glutamine dependent now because they're part of the immune system. And so we can track them down and kill them. We know what their vulnerabilities are. And once you know the biology and biochemistry of stem cells and mesenchymal cells and how you play them off each other, this whole cancer thing, once you understand the biology and biochemistry of the disorder, there are so many nifty ways we can kill these cells. They're not smart. They're stupid. They're actually dependent on a fermentation mechanism, and we know how to stop that. And we're doing everything not to do that. Everything we're doing to these cancer patients is making these tumor cells stronger and survivable. It's just the opposite of what we should be doing.
Right. So there is hope. If we flip it on its head, instead of saying, okay, I was diagnosed with cancer, these same techniques will prevent cancer, right? So if you stay in ketosis, the odds are very small that you'll get cancer.
Yeah, well, our ancestors, those folks who live according to traditional ways, the scientists and physicians who study these folks, they never have cancer. Dogs and wolves never get cancer, or rarely. Domestic cancer is the #1 killer of domestic dogs because they're eating the same crap food that we're eating.
My dog's on pure carnivore, and I have these conversations with my clients, my friends all the time, that he eats completely raw diet.
Yeah.
And I've studied the pet food industry too, and how all these dogs are getting cancers.
Yeah.
Or they're 99% the same as a wolf, and a wolf would never get cancer in nature. Never.
No.
Right?
No,
So-
you never see a wolf attacking a cornfield, right?
No.
They're eating chickens and rats and stuff. Again, it's the diet and lifestyle. And when we looked at African tribes and Inuits and Aboriginals that stayed according to their traditional ways, cancer was extremely rare. They didn't even have a name for it. But now in our great American diet and lifestyle, which is our greatest export to the world, we got cancer, dementia, right? All of these chronic diseases as the result of our diet. Our technology has evolved so much faster than our biology. We are still Paleolithic man living in a new environment. Where we have all these choices that are mostly unhealthy for our physiology and biochemistry. But
Well, Dr. Thomas Seyfried,
as long as we know that, we can do something about it.
and Dr. Thomas Seyfried, I really, really want to honor your time today. I appreciate so much time that you spent with us, and I will be honoring that time by making sure that my patients are definitely utilizing your protocols, trying to get down to exactly what we've talked about today. I look forward to my patients watching this podcast and really benefiting from it. And I can only imagine the millions of lives that you'll be saving with your work from here on out. So really appreciate your time with us.
Thank you for what you're doing. Thank you for talking to us. This is absolutely amazing information. And it gives hope to people that are thinking they don't have a chance.
Yeah, absolutely. And all of our research here at Boston College is supported by private foundations and philanthropy. So there are folks that are excited about this, and they just want to be part of the paradigm change. So Travis Christofferson's Foundation for Cancer Metabolic Therapies, and we have some foundations for breast cancer and pancreatic cancer, actually. So people are throwing money in our direction because it's our work here at Boston College that has started this whole thing, and now we're spreading it around the globe into clinical practice. So that's how it's going to be, how it's going to happen. And we're still trying every kind of a new drug diet combinations to see which one will be the best.
Well, thank you very much. We appreciate it.
Yeah.
Thanks.
You can learn more about Professor Seyfried and his efforts at our website, restorewellness.org. Thank you for listening. We wish you the best of luck in your personal wellness journey.